All right. Thank you all for joining the N of One Collaborative Seminar Series. My name is Nicole Nolan, and I'm N1C's Executive Director. It's my pleasure to welcome you to today's talk. Before I hand it over to our moderator for today, I'll cover just a couple brief notes. The first is that we'd like to express sincere gratitude to the Chan Zuckerberg Initiative for their support of N1C's mission, including the seminar series. Secondly, we'd like to invite you to Connections, N1C's virtual collaboration platform following today's talk. We would love to continue the conversation with you there and invite you to join us. So with those covered, I'd like to hand it over to today's moderator, Jeff Milton, longtime friend and supporter of N1C and co-founder of La Jolla Labs. Jeff?
Thanks, Nicole. Hello, everyone, and we are packing in a lot here, so I'm going to go as fast as I can with the intros, but hopefully do represent everyone well. We want this to be an interactive session, so please, if you can, turn on your cameras um, if it's possible. If you have questions, put them in the chat for discussion after the presentations. And as you might have already seen, this is a recorded session, and you can view it later on Connections. At the end of the seminar, you will automatically receive a survey. So be grateful if you appreciate that if you could give us some feedback. All right, so let's go into the presentations. So the first presenter is, let me get my notes here. And Nicole, correct me if I'm wrong. It's Jake, right? Jake Emerson, right? So first presenter is Jake. Jake is Associate Director of Data Engineering at Jax Data Science. Uh, and he builds fast, reliable software systems, complex data, intensive research workflows. Um, and his background spans software engineering, mechanical, environmental engineering, systems architecture, and scientific software development. Uh, and so today, Jake will share how integrated data infrastructure can support more efficient and reproducible ASO therapeutic development. Jake.
Hey everybody. Yeah, the time is short, so I'll jump right into it. So together with my colleagues Juno and Konstantinos who are on the call, we've been working with the N1C, the JAX Rare Disease Club, Rare Disease Translation Center, and the Sanders Group at Oxford, also with Marlon, who's on the call, and Barney, who will be speaking later as well. And we've been working together on finding ways to bring preclinical data and predictions about ASOs together in a consistent application. And so today, let me get my slides up here. I'm going to talk about, sharing first. As Jeff said, I'm going to be talking about interoperability. And so one of the key things here, of course, get all the bits ready. There we go. Okay, so I'm not going to go deep into the science necessarily, but more into the data organization. At JAX, we have lots of research labs and lots of scientific services, and within our institution, we've had to really fight to find ways to bring data sets together and to make data fair, findable, accessible, interoperable, and reusable. And for the collaboration that we're working on now, one of the key things is the I in fair or interoperability. As we're sharing data across systems, this is a difficult challenge. And certainly since data and tools are constantly changing, we need to find ways to make those new data and tools shareable. And I'm sure that my other presenters today have thought through this to some degree, but one of the things that is really difficult is that as we're working in a really distributed way in research, there's an opportunity for us to share by doing some really fairly simple things, including using ontologies and controlled terms and being consistent in the way that we use our identifiers. And then, of course, using common data sharing formats so that our annotated data can be shared and understood between different disparate teams. This, of course, is not easy. But one of the things that is helpful is that we don't necessarily have to annotate everything, and we don't have to annotate everything perfectly. There's key elements in our different systems that would be shared and need to connect across different systems. And there are great tools out there that we can use and adopt for annotation and data sharing already. So as long as you've got kind of the key concepts in your system and the key language well formalized, you can go a long way towards sharing. So the main idea here is that interoperability means preserving data across institutional boundaries, across study boundaries, across target boundaries, and across ASO boundaries. Different institutions and so on will have different descriptions for these things, but there are ways to make those descriptions be consistent. One really great way to make our data more consistent is just by adopting and applying annotations from well-known repositories. I've got some examples of them here. Variant nomenclature and gene nomenclature are key as target descriptors, but also describing our evidence in consistent ways and sharing data with formats like research object crates and the frictionless data framework are also really helpful in kind of getting over the difficult bars of data sharing. All right, so from there, I'm going to do the dangerous thing and do a live demo. And this is, I'm not going to get into our actual preclinical data sharing database, but I'm going to do a demo just of BioConnect, which is this internal tool that we've developed at JAX as a way to just describe studies. And we're using this as a basis also for the preclinical data in our collaboration with N1C and Oxford. So just briefly, by having our studies, assays, and samples described in consistent ways, and we have a means by which to curate the data and the samples to say what tissue is present, what strains are present for model organisms, measurement types, and the like. And that goes a long way towards just making these data findable in a digital library. And I'll just show you one specific study. One of the things that we found was really difficult was bringing metadata together with the data objects and making sure that there's a clear connection between them. So as a part of our study descriptions, you'll see that there's consistent metadata that's applied to the sources. Move this little thing out of the way. And then within assays, there are samples that have their own description too. And then in order to make this all connect together, the sample identifiers need to match identifiers in the data files. And that's pretty consistent across all of our metadata and all of our data objects. And so then this can be challenging sort of institutionally, but the key ideas are not particularly difficult. So I'm going to show this in what we call a data package. So I've just created this representation of that particular study. I have to keep moving this little thing back and forth. And then I'll download that data package and just very briefly show you what lives inside of there. And this is our core tool for data sharing. So if I open that thing up, you'll see that there are a few different files in here, but the key ones are this JSON file, and I'll open that so you can see the contents. This is a machine readable file. It's a JSON data format that contains descriptions of all the various entities that live in the system. That's a lot of JSON, and nobody loves to read JSON, but I'll show you something a little bit more practical. Whoops. This is the data matrix from that example file. This doesn't look that exciting, but the thing that's really difficult to do, but very helpful to do, is by the time you get to analysis and after all of your assays have been run, it's really common for the metadata to be out of sync with the actual data object. So through through that BioConnect system and through our curation process, we make it really easy for folks to make those combinations. And then for each one of these terms, you'll see and I think this is pretty consistent throughout research. If you have an annotation for data, you might see something like this. There's a measure, some sort of cryptic description at the header of the top of a, column of your data. Um, Between this data matrix, which has all the data and metadata, which is the starting point for analysis, I can also, whoops, I'm going to go back to that thing. I can find out really the details of what is in that column. Control F, V, and if somebody needs to go back and reanalyze these data, body weight is a fairly simple concept, right? But having metadata associated with the time point, associated with the measure, and associated with the specific samples is really it's a fundamental activity for interoperability kind of throughout science, and data sharing generally. So I'm sure we'll find out a lot of more in-depth about ASOs, but for us, data sharing was the main challenge. And I guess I could say my recommendation is that by using shared terminology, especially from public resources, across our various systems, we're able to turn distributed ASO research into connected evidence that supports better comparison, prioritization, and collaboration. So with that, I hopefully am within time, and thank you for your time. I look forward to questions later.
Awesome. Thank you, Jake. And Jason has a question for you. I don't know, Jason, is this Jason Curry? Is this my good friend Jason Curry? If you have any, this is amazing. Are PAGs able to gain access to this? I don't know what PAGs are.
Patient advocacy groups.
Ah, yes, of course you would. Of course, you can educate me more. Yes, it is,
Yeah,
Jason. All
as a
right.
part, yeah, no, thank you. That's a great question. As our project develops, yes, that's our goal, is to make this more broadly used by the community. For now, it's a closed resource. We're opening it up kind of slowly in stages, but we look forward to more users and feedback as well.
This is great. Great. Well done.
Thank you.
Awesome. I have a question. Have you considered using large language models on this to help you do the curation? Because I think that would be a useful tool. Yeah.
Yeah, absolutely. Yes, that's really is our next step. We've been
Mhm.
working on this since well before LLMs were
Yeah.
widely available. But man, they could not have come at a better time, to tell you the truth. I mean, just
Yeah.
curation is difficult and arduous, and if there are ways for
Mhm.
somebody to just take a spreadsheet and upload it and say, curate my stuff for me, then yes, absolutely. And I guess building on that too, for both reanalysis and for meta-analysis, if you, like I say, um, your metadata annotations don't have to be perfect, and that's even more so the case now that we have LLMs. If you have some
Mhm.
basic structure, that goes a really long way towards making data interoperable with LLMs. If you don't have that basic structure, then you're just adding sort of useless variability.
Yeah. Yeah. And underappreciated problem is the curation of this type of data set. I mean, I remember. I mean, it's always been a problem because there's a subjective nature to curation. You have a pathologist's view of it, you have somebody else's view of it, and this can be very hard to bring together. But let's see, any other questions? What was the biggest challenge you faced? This is from Buma Yandava.
The biggest
In,
challenge, I think, really
yeah.
speaks to what Jeff was saying. It's culture. It's getting
Mhm.
everybody kind of on board and behind the idea that you're not curating just for this one paper, but that you're curating for others to reuse your data and your tools.
Yeah, definitely. I totally agree with that. All right. Thank you, Jake. And we will go to our next speaker. And the next speaker is Logan Newton, Sick Kids, Toronto. Logan is a PhD candidate at the Hospital for Sick Children and at the University of Toronto, supervised by Dr. Greg Costain and Dr. Ashish Dashwar. His research focuses on exon skipping and ASO therapy, development of ASO therapies. Logan's group has developed an in silico pipeline to assess the feasibility of exon skipping across a variety of human diseases. Um, in addition to this pipeline, they've developed the Hawkeye database, a web application that supports searching for target exons and optimized ASO sequences. So Logan's going to talk about his in silico design tools uh, and help identify how it can help identify exon skipping candidates. Logan,
Hi, my slides are visible.
your slides are visible. No
Awesome.
notes.
Okay. Hello, everyone. I'm excited to share with you our methods for in silico design and optimization of exon skipping ASOs. So, to induce effective exon skipping, an ASO sequence must fulfill certain criteria. And when comparing to something like a knockdown ASO design, where in theory much of the transcript could be a realistic target, target selection for exon skipping requires very specific and context-dependent considerations. So broadly speaking, an exon skipping ASO should one, bind most preferably to an exonic splicing enhancer or potentially a splice site. Two, the ASO should alter splicing in a predictable way, so ensuring that we minimize unintended splicing events such as intron retention is a must. Three, we have to account for exon-specific biology. So every exon will have variations in where the exonic splicing motifs are, the strength of the splice sites, and the accessibility of the target region, for example. And four, while meeting all these aforementioned criteria, the ASO must also still meet standard sequence and thermodynamic requirements, such as GC content, binding and free energy, and off-target effects. And so luckily for us, when we got started with this, exon skipping ASO design criteria has been very well established, and amazing stepwise guidelines have been published for the design of exon skipping ASOs. And to facilitate each steps of the guidelines, there are many fantastic computational tools that exist, such as BLAST, RNA Structure, mFold, Splice AI, and Rescue ESE. But when we started doing exon skipping ASO design, we found ourselves uploading potential ASO sequences into these tools one by one, assessing the output, referring back to the guidelines, then making a decision for that step. And this can become quite laborious for us. So what we wanted to do was take these tools and these guidelines and really just merge them into one tool. And that one tool being a tool that just accepts one simple input, an exon sequence, and then directly outputs a list of ASOs that meet all established criteria to induce exon skipping. And so we call this tool Hawkeye, the Hawkeye ASO design tool. And we wanted it to be high throughput and scalable so that optimized exon skipping ASOs could be designed for many exons at once. And so our objective is to develop a high throughput tool for generating and prioritizing 20-mer exon skipping ASO sequences that support scalable and proactive ASO design and evaluation. And so this code for the ASO design tool was developed in Python by Bushra Hawk, who's pictured here. And she is our bioinformatician who supports all of the computational work done here at our lab. And broadly, how our ASO design tool works can be broken down into 5 main steps. The first step is ASO sequence generation. So like I mentioned, the tool accepts as input the sequence of a target exon and 20 base pairs into its flanking introns. And then kind of like an ASO walk, we generate all possible 20-mer ASO sequences that could target this exon and its flanking introns. Then what we do is we rank every ASO by its ability to perturb splice site recognition using Splice AI. So first what the tool does is it calculates the baseline Splice AI prediction scores for the acceptor and the donor. And then it sort of does like an in silico masking of the input sequence to mimic an ASO binding, and then recalculates the canonical Splice AI acceptor and donor scores. And then we can calculate the difference in that score between when an ASO is present or not. And from this, we retain the top 100 acceptor and donor hits from our tool. The next step is filtering by sequence composition and thermodynamics. So built into our tool is the algorithm for the existing tool RNA structure. And for each ASO, our tool will then calculate the free energy of the ASO and of ASO complexes, as well as the ASO binding energy. The tool will then also internally calculate things like GC content, melting temperature, and will search for ASOs that contain guanine stretches. ASOs with poor sequence composition or thermodynamics are then filtered out at this step with our tool. And here we are able to remove ASOs that are likely to form stable self-structures or ASO dimers, ASOs that may bind weakly, or ASOs that may stimulate an immune response. Next, we filter by ESE coverage and target accessibility. So like RNA structure, the tool incorporates the existing tools ESE Finder and Rescue ESE to identify exonic splicing enhancers by sequence. So specifically, our tool retains ASOs that significantly overlap SC35 and SRP55 ESEs, as well as rescue ESE sites. And then for target accessibility, we use mFold, and we prioritize ASOs that target partially open and partially closed regions, as in literature targeting these ESEs and mRNAs with this secondary structure has been shown to increase the chances of achieving the intended exon skipping. And then finally, the tool incorporates BLAST to screen for off-target homology and filters out ASOs with 17 or more consecutive matching base pairs to an off-target sequence. Sorry. And then what's outputted by our tool is an Excel file for each exon containing ASO sequences that are in full phosphorothioated 2-mo backbones in IDT format. So you can just quickly paste these ASOs into IDT and order them with ease. And so last year, just to give a bit more context on why we wanted to design this tool and emphasize really its utility, some background about this is, so last year, the N1C patient identification group released the variant guidelines, which outlines step-by-step assessments for assessing variants towards different ASO approaches, and this includes for exon skipping. And this was spearheaded by David Sheary and Marlon Laufer. And these guidelines, having these guidelines permitted us to then create another tool in R that accepts a gene accession number and then pulls from various databases exon level information that allows us to classify each exon with regard to how likely we think that exon can be skipped, then yield a functional protein. So in the context of an ASO therapy, an exon that can be skipped, which we call a dispensable exon, could be bypassed using an ASO to bypass something like a disease-causing nonsense variant. And to facilitate bespoke exon skipping ASO therapy development, what we wanted to do was proactively define exon dispensability across all human disease genes, use our tool to design the corresponding ASOs, and then share this data as an open access web app. And so we assessed 72,644 exons across 5,057 genes. A total of 2,481 were classified as dispensable. So these are the exons that we're predicting with our highest confidence that they can be removed to yield still an in-frame functional protein product. And then using our ASO design tool, we generated ASOs for 2,308 exons with a median of 12 ASOs outputted per exon. And thus far, we have validated experimentally 3 ASOs targeting 3 different genes, 3 different exons that were identified as dispensable. And we just quickly validated them using RT-PCR and semi-quantitative densitometry analysis. So 8 of our 9 tested ASOs did induce the intended exon skipping, and densitometry-based skipping efficiencies range from 21 to 98%. So again, this is semi-quantitative because it's based off of based off a PCR gel. So likely it is an overestimate, but it was nice to see that some of these ASOs we designed using our tool did seem to be working well. And all of these ASO sequences that we have designed are available on the Hawkeye web application. So this is an open access web application that anyone could use to search for a gene of interest. And what it will show for each gene is the number of exons within that gene and the assessment that we've given each exon. And in the table format here, each row will be an exon and its assessment with regards to how skippable it is shown. And it can be flipped through. And then the table can also just be filtered to show ones that we think most likely can be skipped versus ones that we think are less likely to be able to be skipped and will likely result in protein loss of function. The table here at the bottom will display information about the exon that's selected and summarize why it's grouped into that classification. And then if we select a dispensable exon and scroll down, we can download our ASO sequences here at the bottom. As for next steps, with our ASO design tool, we're currently building upon its code to be able to design knockdown ASOs at scale in a similar fashion. So two students in our lab, Rebecca Sakaki and Hui Lin, are working hard on this and hoping to add our knockdown ASOs similarly to the Hawkeye web application. And so, for example, with regards to adjusting our code to be able to do knockdown ASOs, doing things just like removing filtering for splicing impact and splicing motif coverage, adjusting filtering for mRNA secondary structure to instead target fully open regions, and outputting as 510 5 gapmers in IDT format can facilitate instead knockdown instead of splice modulating. So thank you so much. And I have attached here also on the title slide the QR code to access the Hawkeye web app, as well as the link. Thank you.
Awesome. Thank you, Logan. So I have
I have a question, if I might.
oh, sure, go ahead.
Oh,
Yes.
super. Um, yes, thank you. Thank you. That was a fantastic talk, Logan. So, as I understand, your Hawkeye tool seems to be fantastic. I was just trying to do a little bit of, um, I just was trying to use it myself on the side screen, just to see how it is, it can be utilized. One of my questions is, I was just trying to understand your workflow and everything. So as I understand, you were able to identify some 70,000 exons across several, like 5,000 different diseases, all exon skipping, I assume, or exon variants. And then you came down to about 2.5 or 2.8 thousand, I'm sorry, about 2,500 exons. So I'm just trying to understand what that workflow looked like. Like, how did you
Yeah.
move from 70,000 to
Mm, mm.
20,000?
So yeah, sorry, I glossed over some of the background there just for the sake of time. But so we started with 72,000 exons across 5,000 genes. So what those genes are, every monogenic protein coding disease gene in humans. And then we took what our tool does is it looks at each exon across those genes and it ranks them with regards to how likely it thinks that that specific exon could be skipped via an ASO and that the
Mm,
protein can still be functional. So for example, what we can, the labels that we use, and it's on the about page on the website as well, is like something like dispensable, which is a skippable exon, versus indispensable, one that's needed for the protein to function. So the workflow, broadly speaking, and this is also laid out in the N1C variant guidelines, which is what we base this filtering off of. But for example, a dispensable exon would be something like a small in-frame exon that doesn't code for any functionally important domains or PTM sites.
mhm,
It potentially doesn't include any missense variants, instead just coding or having nonsense variants reported. Whereas on the other hand, an indispensable exon would be something like a large out-of-frame exon that codes for multiple functional domains, or potentially contains a start or stop site. And then we also have intermediate tiers between those two of increasingly strict functional domain considerations and size considerations that allows us to have this sort of in-between group. So, for example, we consider things like domain redundancy in those groups. So if there are multiple of the same domain throughout the protein, and that
mhm.
exon codes for just one of them, we consider potentially that that exon might be more likely to be skippable than another. However, because it does still contain a domain, we don't group it into the full dispensable category. So that is just briefly some of our workflow.
Fantastic. Yeah, that explains a lot. Um, and the other question is, so yeah, now getting down to the 2.5, so 2,500, so actionable ASOs, and then you're trying to do wet lab assays, functional assays, I'm thinking, or was it just RT-PCR? So you're coming down to about 10 candidates. And your success rate seems to be impossible. It's very impressive, right? 8 of 9 seems to be rescuing it. So I'm trying
Yeah,
to also understand how that happens, like in the sense, again, how do you go from 2,500 candidates to just 8 or 9 candidates, most of which seems to be successful?
so we had 25 candidate exons that we could skip, what we're saying
Hmm.
might be
Oh,
skippable.
okay. Hmm.
And for each exon, we then designed on average 12 ASOs using our tool.
Okay.
And what I had shown on those slides was we had just sort of picked randomly a couple that we wanted to test. And that
Mm.
RT-PCR gel is not actually testing the function of the protein after exon skipping. What it is just assessing whether or
Hmm.
not our ASO is skipping the exon.
Got it.
Similarly, we have also a few proof of concepts for our tool as well, which I haven't gone over in this presentation, but I will just mention that we have functionally assessed skipping some of these dispensable exons and see an alignment with our tool as well.
Super. And one
Cool.
last question, I promise, if you don't mind. At what step does it does it fail? Like when you're coming down from 70,000 to 8 or 9, which step does it fail usually in silico, when you're in silico designing to
Oh,
get it
like
actually working?
why could we not design ASOs for
Hmm.
every of
Hmm.
other? Yeah. So on the slide, I had mentioned that of the 2,400 exons that we identified as
Hmm.
potentially skippable, we
Hmm.
were able to design ASOs for 93% of them. And that's just because, for example, some exons, when we looked back at the output logs, they were small exons with high sequence homology to other regions in the genome. So, for example, every ASO had off-target effects, or stuff
Hmm.
like the exon sequence was very, very GC heavy, and so the ASOs had very high GC content, so we couldn't design an ASO for them.
Okay,
It's
got
not
it.
using the guidelines we currently, or how we're currently filtering.
Thank you. Thank you so much.
Thank you.
And Bushra,
All
thank you
right.
for the link.
We got a couple other questions, and then we'll jump to the next talk. So, uh, the, what about poison exons? Did you get a chance to look at this in the context of poison exons?
Oh,
Um,
we have not looked at it in the context of poison exons, but that's a great point. Definitely should.
yeah. Okay, and then let's see. Intron skipping ASO is a thing. For when an intronic mutation leads to a splicing acceptor creation. So I guess this, I'm looking at this in the uh, incorrect inclusion. Um, so
So I don't
yeah,
for
go
that.
ahead.
I don't think the term it's not like really intron skipping. It would more so be
Yeah.
masking the cryptic splice site in the intron to prevent
Mhm.
its usage, just sterically blocking it to then increase the use again of the canonical splice site. So that's definitely a thing, and we definitely
Mhm.
see that in a lot of ASO work is that sort of splice correcting ASO.
All right, then for Marlon, exon splice silencers. Have you looked at those? Uh,
We had initially we are what are in a specific stage of our tool, we had oh, mapped where exonic splicing silencers were, and we thought about incorporating a way to avoid exonic splicing silencers, like filter out ASOs that overlap ESSs. Um, Ultimately, it brought down our number a lot, and we thought that a median of something like 12 ASOs being outputted per exon was a good starting point, and we didn't really want to filter it down too much by avoiding ESSs.
Cool. Awesome. Logan, great talk. That's definitely I think there's a lot of exciting work going on in the Exxon space and it's really cool to see. This is very, very efficient tool. So I've seen that from definitely from experience. So okay. So we're gonna move to our next speaker. Let's go to Barney Hill, University of Oxford. And Barney is a statistics PhD candidate at the University of Oxford. He's supervised by Carla Rinaldi, Nikki Whiffen, and Stefan Sanders. His work focuses on computational approaches to ASO safety and preclinical evaluation. He'll introduce ASO Atlas 2.0, a data set covering the GAPMER ASO preclinical pipeline. It's a data set includes in vivo hepatotox, neurotox endpoints. This is all stuff that's come out of the Ionis. A lot of Ionis patents is, basically has a machine that patents everything that I partially built. So I'm partially responsible for causing this mess. But actually, it's good. It's all good. It's all public data, so it's good. We can now scrape it and get some good machine learning out of it. So. So Barney's going to talk about some predictive modeling. that can help us make smarter decisions when it comes to these types of designs. So, Barney.
Thank you so much, Jeff. Can you see the slides that are shared?
All good. Yep, looks good.
Awesome. Yeah, so today I'm just going to be sharing a kind of collection of things that we've been thinking about recently in relation to ASOs and N of one. So kind of going back to basics, I think when we think about like innovation in the context of gamma ASOs, we typically think of this kind of chemical modifications, progressively improving clinical efficacy. I think what's becoming increasingly clear, though, is that there exists a large body of patients with neurological conditions who, are likely in some way amenable to ASOs beginning from second generation MOE chemistry, and yet the cost just remains too high. So we're kind of focused on cost and reducing the preclinical cost, and I think the primary way we believe we can achieve this is through data and modeling. Over the last year or so, we've invested significant amount of resources into the collation of the Ionis preclinical patent data. Ionis has published over 280,000 measurements across over 400 target genes. These target genes are grouped predominantly in the, I would say, neurological conditions. And our new tranche of data, we're not just releasing, this in vitro hit screening data. We now have in vitro dose response data, and crucially, quite large amounts of in vivo neurotolerability and liver toxicity data. And the brilliant thing about this data as well is it's all linked together. Well, not all of it, but for a large number of compounds, we can observe the kind of measurements at multiple stages of the pipeline. Previously, we investigated in vitro efficacy and the factors affecting it. We identified that the position of certain nucleotides affected efficacy. So in, I think, orange, you see a kind of negative impact. These Cs present in 310-free constrained F4 have a negative impact on efficacy. And quite surprisingly, we saw this pattern totally reverse in another chemistry. And so we believe chemistry is important alongside sequence when you're modeling ASOs. We also observed that target context matters a lot. So you can see that exonic regions are much more enriched for higher knockdown ASOs. And so I think what we learned here is that a lot of things go into ASO in vitro efficacy. There's not just one factor. And so we built a model to kind of account for as much of this as possible. We modeled RNA context sequence, the ASO itself, some of these chemical modifications, and then the experimental factors, including dosage and transaction method. We went to test this in a lab, so we designed a set of 5-10-5 MOE gammas against KCNT2, which is a disease associated with early onset epilepsies. As a standard, we just randomly screened across the transcript, manufactured those ASOs, but we also manufactured a second set of ASOs, which were prioritized by oligo AI. And in our experiment, we identified that in this particular context, this is an unseen gene, oligo AI was capable of approximately 5-fold reduction in screening effort. So this is all very well and good, but my initial objective was really to reduce costs, and it's not clear at all how in vitro, the reduction of in vitro screening reduces cost. And so the following work is an effort to really quantify this in a measurable way. So we went back to the Ionis data, and we observed that Ionis appear to be publishing data in a very unbiased way. So, regardless of whether there's a negative or positive outcome, you can see the distribution is relatively even. And so we're going to use these distributions to make statements on, given my target in preclinical, say I want inhibition above 80% at a given dosage, what proportion of ASOs are passing these thresholds? And so we repeated this for some of these mouse tolerability metrics, ALT, very classic we want our mice having below double the upper limit of normal. And the same for functional observation battery scores. We want a very low score. And we can repeat the same for rats. And finally, these kind of monkey values for which, at this point in the pipeline, there are very few negative toxicity outcomes. And so, yeah, what I want you to appreciate of this is that this is not like a real pipeline. Ionis does not do this in-house, but it's kind of the best we could do. I hope you appreciate that. And so now that we have, success thresholds at every single point, we can say, well, we want to end up with one clinical candidate. We can then use these passing thresholds to create estimates of how many ASOs we need to screen at each stage to end up with our final clinical candidate. And so once we have this kind of model of how big our pipeline should be, we can then introduce these costs. So we talked to a number of CROs and assembled these very, very rough estimates on what each stage costs for each ASO. These numbers will be different to you, but we hope you can kind of plug your own numbers in and use this for modeling your ASO algorithms. What you can kind of do then is say, well, given if we're using oligo AI with its 5 times better predictions, well, this is actually having a very marginal impact on the total preclinical cost. This probably isn't even worth doing. And so we began focusing a lot on in vivo toxicity and modeling the factors of in vivo toxicity. So I think as has been observed previously by a number of authors, the dinucleotide content of the ASO is actually a very good predictor for some of these toxicity measures. I think Peter Hagedorn a few years ago kind of released this paper showing that these presence of these guanine strings have a very strong correlation with neurotoxicity and acute neurotoxicity. And so we have, just very basic models trained on these dinucleotide inputs, and we get quite good enrichments, around 1.5-fold enrichment of liver toxicity and 2-fold enrichments of neurotolerability. And then we can place these within our pipeline and say, well, given the increase of success rates at our stages, what is the corresponding decrease in cost? So we can go take a model and say, okay, this is the actual cost impact. All of this, all of the paper isn't released at the moment, but you can try out some of these toxicity values now. I think, we just updated Oligo AI, so you can do things like look at neurotoxicity, liver toxicity. We have off targets. You can see whether the gene is present in lots of different species. We're releasing like allele-specific tools. So you can upload someone's genome via the FASTA file, and you can see, given their 2 haplotypes, what are the kind of potential ASOs that can achieve allele-specific effects. And thank you very much. The initial in vitro work is available publicly, and hopefully the in vivo work is available very soon. Thank you.
Fantastic. Thank you, Barney. Reducing the preclinical cost is the holy grail for this whole industry, definitely. And it's very hard to measure. It's very hard to translate cell data into the clinic. One of the assays that actually shows pretty good correlation is the BJAB assay. Did you integrate the BJAB at all in these studies, or have you sort of thought about that as far as have you guys ever used that?
Yeah, I mean, I always say this is like the assay I most want data on, and there is no data on it. I think Ionis have like a dozen released sequences of BJAB assays.
Okay,
So if
we
anyone in
should
this
talk.
school has data,
We should talk. We have a lot of BJAB assay, so we do that. We do that a lot. So happy to kind of merge our data set into that. See what you get out of that.
for sure.
So there is a quest if you have that publicly available to drop the link into the chat. And then
Uh,
go ahead. Did you want to say something about access to the tool?
yeah, it's kind of under review at the moment. I can probably share a link in the chat later. Yeah. But as a kind of very rough form of the data set, I can share.
So I have one more question, unless somebody else wants to jump in. But I have a question on steric blocking. Did you look at steric blocking ASOs? So they sit fully modified ASOs. You were talking a lot about gapmers, but as far as toxicity goes, what was there a difference between the fully modified ASOs and the gapmer ASOs? Did you see anything there?
Yeah, unfortunately, I think there's only around like 5,000 steric blocking ASOs released by ONUS and their patents. So we
I
didn't go
see.
after that, but for
Okay.
sure, someone good.
Yeah, it would be interesting to see that. And I thought it was very interesting to see that there wasn't a lot of toxicity um, in the non-human primates.
Yeah,
So I think in my mind, I'm thinking there's so much toxicity associated with the small volume with mice and rodents that when you dose, it's just such a massive hit. I just think
yeah,
there's going to be so much more statistically. There's going to be so much. I mean, it's probably a good filter in that sense, but I think there could be some confounding variables there that we're dealing with. That things are not as toxic as they actually are. And even
for sure.
Spinraza killed a few mice in acute
Mm.
neurotox. So it's hard to say where we are with that. But any other questions? Awesome. Fantastic. Thank you, Barney.
Thank you.
We'll go to our last talk now. I'm sorry, we're a little bit a little bit slow. Okay. So I'm going to keep this brief. Emilio Harris Mostert at is a PhD candidate at Professor uh, Algarisma's group in the clinical genetics department at Erasmus Medical Center. His background is in life science, technology, and neuroscience. Um, a member of the Dutch Center for RNA Therapeutics, has focused on developing ASO therapies for rare neurodevelopmental disorders. Within the ASO field, Emilio studied allele-selective gapmers and works to improve in silico methods for gapmer targeting. And today he'll introduce Eraser, a tool that evaluates RNA-dependent ASO target sequences across a gene. So with that, Emilio.
Hello. Um, are you seeing my screen or my face?
You're good. We see the
Perfect.
the
Okay,
slides.
perfect. Then, hi, I'm Emilio, and indeed, I'm going to introduce Eraser. And Eraser is a platform for selecting gapmer ASOs. Because to define the designing process, like ideally, it would be like a shotgun approach where you just design as many targets as you can get and find out which ones work. But that's quite time-consuming and expensive. So you kind of have to go for a two-phase design where you select a couple of sequences, you see which ones show any activity, and then you have to kind of go more in detail within those sequences. So what you would like to do is to kind of select the right sequences for this two-phase design. And for that, you need a more detailed approach where you really go into detail on every single sequence that you are going to select. However, to really do this by hand is really time consuming. So we set out to kind of make a tool, which we call Eraser for Erasmus ASO Selector, because we're at the Erasmus Medical Center. And this tool would help us select these ASOs by doing this detailed approach for us, where it generates all possible gapmer ASOs for a target gene, and then it filters these ASOs based on input that we want. And these filters are based on characteristics that are important for ASO functioning. And with this, we accelerate the first selection of promising ASO targets. Of course, this is not the first tool. There are some ranking tools, just like OligoAI, that were introduced. And these are quite nice, and quite a few of them actually work quite well. But the thing is that it lacks information on the given ASO. So we set out to make a different kind of tool, which is more based on the information. Also there are a few tools like ClinASO or ASOG. However, with Eraser, we want to add quite a bit more information for each sequence, and we have a user interface that we think works quite nice as well. The starting point for Eraser was a different script, which was actually made by Lieke Pedersen and her colleagues. And there they already did quite a few stuff. So they looked at secondary structure, at the mouse conservation, at polymorphism frequency, and some basic off-target filtering. So we expanded on that with, well, first of all, a user interface, but we also added a lot more information about these ASOs and some filtering steps for that. And we also expanded it to also design sequences for these patient-specific sequences to make allele-selective ASOs for SNPs that the patient has. And we have an input where you can actually validate your own ASO. So if we look at the main interface of Eraser, here on the left, we have our input part. And in this main input, you put in your gene and you put on whatever filters you want. And that brings you to the first mode for Eraser, which is just a general knockdown. And in this general knockdown, you get kind of three output tables. And the first one is the main results table, where we look at different sequence features, like GC content and that type of stuff. Some information on secondary structure of both your target mRNA, but also of the ASO itself. And some information on the off-target profile, where we try to give it a score based on the counts, but also the risk associated with every off-target. And if you then select one of those ASOs, you go to the off-target page. And in this off-target page, you get detailed information about which off-targets there are, what the mismatches are between your ASO and that off-target, but also, for example, where these off-targets are expressed so that you can get an idea how risky is this really for the ASO that I'm developing. And then finally, we have an RNase H page where there's some information on how RNase H would likely cleave this sequence that you can use to optimize your design. And then finally, we go to, well, the next mode, which is really the patient-specific design. And in this case, we have a VCF file as input for your design start. And this is a file that contains the variants that someone has, that a patient has, or just an individual. And first of all, we get a summary of the variants that are found, so all the SNPs in the gene of interest. And then Eraser will design ASOs targeting these SNPs, but it will also design general ASOs that target the gene of interest. And if this data is phased, the ASOs that are designed will really be allele selective for the allele that you would want to knock down if your patient has a heterozygous gain of function or dominant negative mutation. So what that looks like is here in the patient specific design page. The input is still the gene of interest, but then here you input the VCF file of interest, and then you still have the filtering options, just like in the general knockdown page. And then we move on to like the information that comes out. So here we have just the gene of interest and like amount of variants, and then just a summary of all the variants that are found. And then in the next page, which is the results page, we see the ASOs that are designed with this method, and then also like what the mismatches would be between your on-target, your ASO targeting your on-target, and then the off-target allele that you would not want to target. And then finally, we have our specific ASO input. And in this input, you get to input ASOs of interest, and then Eraser will compute all the information you want for these ASOs of interest. Of course, you have to test this, so we're slowly getting started with that. And this is one example where a student of our lab wanted to knock down MINC1. And we wanted to knock down both the human and the mouse gene for this target. And if you look at MINC1, there are 40,000 possible ASOs that would knock this down potentially. So we ran Eraser, it ran for 22 minutes, and we filtered it down to 120. Because it was just like initial idea, we wanted to see like how we knock this down. We only ordered 3 of these and tested them. And when we test these on human cells, we see that 2 out of 3 had at least some knockdown. And if we test these on mouse, and we only test the first 2 for now, we also see that these 2 have knockdown. Which means that for now, 2 out of 3 ASOs designed with ASO show efficacy. Now, of course, we need a lot more validation and testing for that, but we felt this was a pretty good start for a really moderate first experiment. So in summary, with Eraser, we can really speed up the design of CAPMAR ASOs with a user-friendly interface that will make it a lot more time and cost efficient. We are still implementing some new stuff. For example, that you can just input a custom sequence so that ASOs will be designed for that. And it will be public available, hopefully pretty soon. If you want to help us test it and everything, please send us a message because I think within the next few weeks we will have a version available that we would like to send around to some people for testing. So thanks a lot, first of all, to Thierry, who is sitting next to me and who is the master student who is currently working on the project. I also want to shout out some people from the Hogeschool Leiden because they really coded the groundwork for this script, and Clemens Logman in Tübingen, who kind of made the basis of our off-target search. So I'll leave this up for scanning the QR code or writing down our emails, and I'm happy to answer any questions.
Awesome. Thank you, Emilio. Very efficient talk for a very efficient tool. So that's fantastic. There was we had a couple minutes for questions. So there was a question about, have you validated the off targets with RNA-seq data?
No, not yet. We're really
Okay.
like we'll get that soon, but we're still testing the tool itself. But we are definitely going to be doing that. Yes. Yeah.
Cool. I have a small story about off target. A long time ago, there was a company called Centeris that created an LNA gapmer for androgen receptor. We ran our off targets on that and found that androgen receptor, they knocked down this androgen receptor, but they also knocked down MCM9, which is a DNA repair gene. So it was a cancer drug that actually made it all the way into the clinic and failed in the clinic. And it could be because it's actually knocking down a DNA repair in addition to the androgen receptor. So, kind of, if they had done their off targets, it would never have made it. So it's amazing. Back in those days, maybe the whole genome was probably a different draft version or whatever. But in any case, let's see. We are 3 minutes. So last question. Let's see. Last question. How is neural tolerability of gapmers analyzed? So that's a broad question, but maybe Amelia, if you want to take that from the in silico perspective.
Yes. Yeah. So we have the scripts from the Hagedorn et al. Paper for the acute neurotolerability. So that's in there, and you get the score out there as well, and you can also filter on that if you want. So that's the one thing I think at the moment that we can model, and we do have it in there. Yes.
Cool. Yeah, and I can tell you from experience, when I was at Ionis, we were able to do a pretty good acute tox predictor. The delayed tox was nowhere. We just didn't have enough data, and there just isn't enough data out there, so it's a little harder to do that. But acute neurotox, in many cases, can be predicted. Um, for Gapmers, I mean that's all we did was 5-10-5 Gapmers. And by the way, if you're not familiar with the chemistry, the CET chemistry is really kind of an LNA. It's just a slight change of an LNA chemistry. It's kind of the Ionis version of an LNA. So when we talk about Gapmers, CET Gapmers, 310-3 CET Gapmers, you might as well kind of think of that as a 310-3 LNA Gapmer. For patient-specific design, did you me and you compare? Oh, oops, I can't read that. One second. Comparison with patient VCF and general for efficacy. Let's see.
Yes.
Yeah, go ahead.
Yeah. So we like what the tool does at the moment is so with the VCF file, which contains all the variants that someone has in their genome. So what it does is that for the gene of interest, it takes all of these variants out there, and then instead of taking the reference genome for the sequences around that SNP, it will then take the variant that is found in the VCF file, so that you kind of implement that variant in your ASO designs immediately. And then if it's phased, you can also take a separate which phase is the variant, the pathogenic variant itself on, and then you can actually design ASOs targeting specifically that allele. We don't yet have any further things that would design which of these gapmers targeting the pathogenic allele will actually be more selective. But it is something we're thinking about. But I think for a first version, it will not contain it, but that might be later on.
Thank you.
Awesome. Thank you. And thank you to all of our speakers. Amazing tools, amazing presentations today. And we are at time. So perfect. Thank you all for coming.
Thank
And
you,
please fill
Jeff.
out the survey. Yeah.
Thank you for stimulating great discussion and keeping us on time. I'm very impressed. 12 o'clock exactly.
Perfect.
Thanks, everyone.
All right.
Bye bye.
Bye-bye.